First approach is molecular docking. Second approach is de novo drug design. When we, the main basis for doing structure-based drug design is, it depends upon the 3D structure of the target. When we know the 3D structure of the target, then only we can go for SBDD. So the word is de novo means from the beginning, indicating that with this method, one can generate novel molecular entities without starting template. Computational de novo drug design means designing of novel compounds from scratch, building them directly inside the binding site of the target macromolecule piece by piece. I will show the diagram, then you can better appreciate. De novo drug design refers to the design of novel chemical entities that fit a set of constraints using computational growth algorithms. So this builds, actually these are the diagrams. So this is the binding cavity. What we do, we space the chemical and we place the chemical entities into the each pocket and will grow, like here the pink color, sorry, this one is growing, building block, we call it as a builds the new by initial seed will be ligand growing, ligand linking approaches. Here ligand is growing with pink color, that is pyrrole, then and in another cavity benzene growing, then OH like. So with the ligand growing method, the functional groups are added to the chemical handles of the core fragment. We have to add the core fragment will be placed in the binding cavity. Then the ligand is growing method is done by adding functional groups. The user can therefore pick the most interesting functional groups from the list and add to the chemical handles of the core. De novo drug designs, generating new compounds. That's why de novo drug design. A series of novel small molecular inhibitors of cyclophilin A were identified using de novo drug design approach. The de novo drug design of ligand is performed by building each compound. So this is the diagram. I don't know how to do the annotations. Okay, I hope you are understanding. Within the active site. On the basis of extra structure of target protein, compounds are built within the binding cavity using number of fragments. So joining the fragment linking, ligand linking, ligand building, all this is done. De novo linking. So here also we can say after placing the chemical entities in the binding cavities, the fragments, chemical entities are fragments, then they are linked with the red color link. Okay, then you will get the finally designed compound. So small building blocks. This method starts with the small building blocks are nothing but chemical molecules. The initial molecular building blocks with desired properties are either elaborated on, so we can grow. Directly molecules are connected, ligand linking, ligand building. Successful application of this approach was angiotensin receptor 2 antagonist, valsartan. So this is the application by de novo linking and de novo evolution. This method searches novel ligands by using 3D structural information of protein target, which are usually, so actually this entire construction of the ligand is done within the binding cavity or binding site and calculated by the interaction energy of the target protein with the ligands. So by using fragment-based de novo ligand design, it is possible to assemble molecules that are drug-like, which with much less space. So we have to keep in one mind that there should not be a much larger space. Such a space should be less, having to be explored, which is to be explored. Limitation, high complexity. In some cases, it is less successful in generating drug candidates compared to other methods such as virtual high throughput screening methods for large databases. Here for de novo, the example is design inhibitors for E. coli RNS polymerase using the de novo drug design program SPROUTS. In another study, the SPROUT program, novel inhibitors were developed for Enterococcus faecium ligase VAN-A using hydroxyethylamine as the base template structure. These are the examples of the programs for de novo. So everything, softwares are there, tools are there, programs are there for doing all these studies, binding affinity, binding, building, ligand linking, all that. LigMerge, AutoGrow software, SLICS, Link Builder, and Bomb. So these are the softwares because we have to build in the binding cavity. For that, we have to either link the ligands or chemical entities or build the molecules by connecting. Is available. Link Builder is available for download. So this is very important. Here the ligands are either grown or linked by the user's choice. An empirical scoring function is used to estimate the binding affinities. So the next one is BOMB, Biochemical and Organic Model Builder. Ligand growing programs can be used to design the ligands and that bind to the target without using ligand databases. Here we have to use the fragments. It is used to grow molecules by adding substituents into a core structure. Do you know what drug design? So successful applications under this approach is nelfinavir is the first non-peptidomimetic HIV-1 PR inhibitor to receive FDA approval. De novo, so this is HIV-1 PR inhibitor, applying de novo inhibitor design is in combination with MD simulations led to the discovery of zanamivir is the first new class of neuraminidase inhibitor targeting influenza. Vir means antiviral. Anyway, you are all aware. So next is structure-based virtual high-throughput screening. So after the two approaches in SBDD, that is molecular docking and ligand-based, the next computational is structure-based virtual high-throughput screening. So in this virtual high-throughput, we have to store the databases. I already said PDB, Zinc, all the databases will store the 3D structures along with the properties. Then the target is squeezed against the library of drug molecules. After screening, the binding affinities are estimated by scoring functions. So successful application is here is used identification of inhibitors of protein kinase C2, anti-tumor drugs targeting. So why I'm discussing all these successful applications? Because we think that by virtually how many drugs we got, only one or two. Virtual is not, but virtual along with the dry lab with the wet lab is necessary. Even though we get the drugs, but we have to perform the clinical studies. This is just like a computational is just like a map. With the map, we cannot get the accurate results, but the leads can be generated. Of course, we will get the leads. So that lead optimization can be done using ADME studies. Why lead optimization is done? Because to enhance the effectiveness of promising hits, to obtain the desired pharmacological profiles, to reach the required affinity, pharmacokinetic properties, drug safety, and ADME properties. So by increasing the affinity of the target, its potency can be increased. Then the successful application here is docetaxel. Is an anti-cancer molecule. It is used to treat various types of cancers. In this study, solubility parameters for DTX were obtained using MD molecular dynamic simulations. This in silico model was in agreement with the experimental solubility. So it is very interesting. The experimental solubility of DTX. These are the tools: QuickProp, WallSurf, FF. These are ADME tools which determine the physically significant descriptors and study the behavior of novel molecules, ADME properties, ADME and toxicity filtering tool which calculate physicochemical properties, toxic and unstable groups. These are the tools for ADME study, property study. So next is after structure-based drug design, then we'll talk about the second approach, that is ligand-based drug design. So this is alternative protocol to SBDD approach. Actually, when we don't know the